In a cramped London lab in 1928, Alexander Fleming noticed a round patch of mold and a clear ring where bacteria had failed to grow. He shrugged it off as a curiosity and left the dish on a bench. About a decade later, Howard Florey and Ernst Chain turned that curiosity into penicillin, and the world began to change. But after nearly a century of success, a new danger has emerged. Bacteria are fighting back against those drugs, and time is running out. To understand why the race matters, we have to step into a world before antibiotics. Hospitals in the early 20th century smelled of carbolic soap and iron. Surgeons stitched wounds knowing that a small nick could spiral into fatal sepsis. Childbirth was often a gamble, and simple infections could mean weeks of fever or even an amputation. Alexander Fleming's serendipitous observation of Penicillium notatum at St. Mary's Hospital in London in 1928 planted a seed, but he did not have the chemistry or the yields to turn that seed into a medical revolution. In the early 1930s, Gerhard Domag's discovery of Prontosil gave medicine its first real chemical crack at bacterial disease, though its reach was still limited. The breakthrough that changed everything came at Oxford. Howard Florey, Ernst Chain, and their colleagues purified penicillin in 1940, and in 1941 treated a gardener named Albert Alexander at the Radcliffe Infirmary. His brief recovery showed both the promise of the drug and how scarce it was. The next leap was industrial. Andrew J. Moyer at the USDA improved deep tank fermentation in 1943, and by 1944, firms like Pfizer and Merck were able to mass produce penicillin. Surgeons on the battlefields watched infection rates fall. The 1940s and 1950s became a golden age. Chemists created whole families of drugs, beta-lactams, cephalosporins, carbapenems, aminoglycosides, tetracyclanes, macrolides, and later oxazolidinones and lipopeptides. Each drug targeted a specific bacterial machine: the cell wall, the ribosome, DNA gyrase, or folate metabolism. For the first time, routine infections became reliably reversible, and white coats brimmed with confidence. But that confidence came with a caveat. In his 1945 Nobel lecture, Alexander Fleming warned that careless use could select for resistant organisms. That biological principle sat quietly at the heart of every triumph and set the stage for an escalation that would test medicine and society alike. The first sign that the miracle might have a cost was surprisingly mundane. In hospital notes and lab logs from the late 1940s, clinicians began to report staphylococci that survived penicillin. In the fluorescent glare of post-war wards, microbiologists watched colonies persist where sterilization should have killed them. A few decades later, the pattern hardened. Methicillin was introduced in 1959 to evade penicillin-destroying enzymes. But within 2 years, isolates of methicillin-resistant Staphylococcus aureus were described in the United Kingdom in 1961. The scene was ordinary. A post-operative wound refused to heal, a fever climbed, and the empiric penicillin that would previously have settled the infection failed. The hospital had to isolate the patient, contact tracing became urgent, and public health notices circulated. Resistance was no longer an abstract warning, it was a patient in a bed. On the research bench, families of enzymes emerged that could destroy antibiotics. Beta-lactamases were the simplest and most devastating. These molecules cleave the core ring of penicillins and cephalosporins. Over time, some of these enzymes evolved into what we call extended-spectrum beta-lactamases, and families like CTXM began to topple third-generation cephalosporins. Plasmids that carried those enzyme genes traveled freely between species in the crowded gut and on hospital surfaces. Researchers also began to see other escapes: alterations in drug target structure, changes in membrane permeability, and the rise of efflux pumps that literally spit drugs back out. The interplay between hospital practices, heavy-handed empirical prescribing, and the agricultural use of antibiotics as cheap growth promoters created a global laboratory of selection. Pressure built steadily, and the early victories of medicine now had to contend with bacteria that were learning to survive. To see how bacteria adapt, we need to zoom down to the molecular battlefield. The beta-lactam class of antibiotics attacks penicillin-binding proteins, the enzymes that stitch peptidoglycan into a rigid cell wall. Change the lock and the key no longer fits. The MEK-A gene, for example, encodes a surrogate enzyme called PBP2A that makes many staphylococci resistant to classic beta-lactams. That single genetic shift is what turned methicillin-resistant Staphylococcus aureus into a household name. But enzymes are not the whole story. Some bacteria produce aminoglycoside-modifying enzymes with names like AAC, ANT, and APH, acetyltransferases, nucleotidyltransferases, and phosphotransferases, which chemically alter drugs so they can no longer bind the ribosome. Tetracycline resistance often comes from ribosomal protection proteins such as TET-M, which physically dislodge the drug. Macrolide resistance frequently involves ARM methylases that methylate the 23S ribosomal RNA and block macrolide binding. Fluoroquinolone resistance commonly arises from point mutations in GRA or PAR-C, or from plasmid-encoded QNR proteins that shield DNA gyrase. Porin loss is a clever physical defense. Enterobacteriaceae can downregulate or mutate outer membrane porins, and Pseudomonas alters porins like O/D, effectively closing gateways that once ferried carbapenems inside. Then there are efflux pumps, Acrab Tol-C in E. coli or the MEX systems in Pseudomonas, which act like microscopic bilge pumps, extruding diverse drugs. Horizontal gene transfer amplifies the problem. Integrons, transposons, and conjugative plasmids assemble resistance gene cassettes and shuttle them across species and geographies. KPC carbapenemases, first described in the 1990s, NDM1 reported in 2008, and plasmid-mediated MCR1 for colistin in 2015 are concrete milestones showing how mobile these threats can be. Clinically, the consequences are stark. Bacteremias due to carbapenem-resistant Enterobacteriaceae have been reported with mortality rates as high as 40 to 50% in some series. Vancomycin-resistant Enterococci complicate transplants and chemotherapy. Extensively drug-resistant tuberculosis requires months of toxic therapy. All of this happens while diagnostic gaps delay targeted treatment and empirical broad-spectrum use feeds further selection. So the problem stops being purely biological and becomes a systemic crisis, spanning farms, clinics, wastewater, and supply chains. Understanding these mechanisms matters because it tells us where to intervene, how resistance spreads, and why coordinated action is essential. By the early 21st century, the tension had reached a new pitch. In intensive care wards, a ventilated patient with a bloodstream infection might be carrying bacteria resistant to penicillins, cephalosporins, carbapenems, and even last-resort polymyxins. Colistin, once shelved because of toxicity, had become a drug of last resort. And then in 2015, scientists in China reported a plasmid-mediated MCR1 gene, showing that even colistin resistance could travel between bacteria. The map of outbreaks expanded and went public. KPC producing Klebsiella in the United States, NDM1 linked to travel and healthcare exposure in South Asia, Oxa-type carbapenemases appearing in Europe, and endemic multi-drug-resistant Acinetobacter in conflict zones. The arms race against bacteria collided with an economic impasse. Big pharmaceutical firms found antibiotics unattractive as a business. The return on investment fell far short of what chronic disease drugs could deliver, and many companies scaled back or shut down discovery programs. The World Health Organization, and then the Independent Review on Antimicrobial Resistance, led by Jim O'Neill in 2016, began to speak bluntly. Without decisive action, antimicrobial resistance could cause up to 10 million deaths per year by 2050. The media amplified that apocalyptic frame and politicians started to act, but funding and coordination lagged behind the rhetoric. Environmental stories added urgency. Researchers walking along rivers below drug manufacturing sites in India found high concentrations of active antibiotics downstream. The water sometimes smelled faintly of chemical effluent, and bacterial populations carried multiple resistance genes. The human reality was immediate and painful. A parent in a low-resource hospital watching IV lines and monitors beep while clinicians offered combinations of toxic drugs with limited evidence of benefit. The old model of discovery followed by deployment was exhausted. What was needed was structural change, better diagnostics to target therapy, stronger stewardship to slow selection, new economic incentives to revive research and development, and alternative strategies to treat infections that no longer respond to classic antibiotics. The turning point did not arrive as a single eureka moment in a lab. It came as a series of high-stakes demonstrations showing that a different strategy might actually work. In 2015, a paper describing Teixobactin, discovered using an iChip platform, showed that soil bacteria harbored molecules active against gram-positive pathogens and suggested a pipeline beyond conventional culturing. Around the same time, and starting in 2014, researchers demonstrated programmable antimicrobials using CRISPR systems to target and cut resistance genes, opening the idea of sequence-specific bacterial killing. Then in 2016, the dramatic clinical rescue of Tom Patterson from a nearly untreatable Acinetobacter infection with intravenous bacteriophage therapy captured worldwide attention. His wife, Stephanie Strathdy, pulled together teams from UC San Diego, Baylor, and the Eliava Institute in Georgia. With an emergency investigational authorization from the FDA, a tailored cocktail of viruses that infect bacteria was prepared and administered, and within days, Patterson improved. It was a proof of concept. Alternative biologic therapeutics, combined with rapid genomic diagnostics, could save lives. At the same time, regulators and funders began to move. The GAIN Act had already created incentives for new antibiotics. CARB-X launched in 2016 to fund early-stage projects, and later efforts like the AMR Action Fund marshaled public-private capital to shore up the pipeline. New agents reached patients. Ceftazidime avibactam tackled resistant Enterobacteriaceae, and cefiderocol, a siderophore cephalosporin, earned approval for difficult gram-negative infections. The immediate consequence was a shift in narrative from inevitability to contested possibility. Laboratories that once only produced antibiograms began offering rapid PCR panels, MALDI-TOF identification, and whole genome sequencing to steer therapy. The crisis that had felt unstoppable now had credible countermeasures, and a new playbook began to form. The combined scientific, regulatory, and policy response has changed how medicine confronts bacterial disease. Hospitals now routinely run antimicrobial stewardship programs that audit prescriptions, promote de-escalation, and favor shorter, evidence-based durations. Public health agencies from the CDC to the World Health Organization have published priority pathogen lists and action plans, and some countries have piloted subscription-style payments to help companies sustain antibiotic development. The rise of rapid bedside diagnostics is shrinking needless broad-spectrum exposure, and genome sequencing now tracks outbreaks in near real time. Industry has re-engaged, aided by push funds, prizes, and novel reimbursement experiments, but the market remains fragile. Environmental regulation of pharmaceutical production and restrictions on antibiotic growth promoters in agriculture, especially in places like the European Union, have also reduced some selective pressures. The lesson is systemic. Antibiotics are not just pills, but public goods that require stewardship. Stewardship means better science, smarter diagnostics, and durable economic models. The race that began with Fleming's mold is far from over, but the pivot we have witnessed shows that with coordinated science, policy, and clinical will, we can build a resilient set of tools and tactics to keep infections treatable for generations to come.